Hi, this is Miss Litton. It's still my period 1 class. Say hi. All right, so we are on chapter 19, continuing on. We did 19.1, systematic biology. We are on 19.2, the 3 domain system. And so the first thing I want to do is defining domains. You are comparing rRNA sequences. Can show that bacteria and archaea branched early from Luca. Who is Luca? Nice. And then eukarya branched from archaea. I gave you a little side note there about ribosomal RNA mutates very what? No, I didn't say quickly. Ribosomal RNA mutates very slowly. So when you compare rRNA sequences, where do you find rRNA? In what? Ribosomes. What happens on ribosomes? Protein synthesis. Okay. So rRNA nucleic acid sequences mutate very, very slowly. So by comparing those sequences, you can see when you diverged from your ancestral species, and you can look in the hundreds of millions of years. Conversely, where else can you find DNA? Or nucleic acid, you can find it in the nucleus. Where else? Mitochondrial. Mitochondrial DNA mutates very rapidly. So that's if you're trying to distinguish between closely related species, you can use mitochondrial DNA. So, pardon?
Chloroplast also, right?
Yeah, if you had it. Yeah. Okay, so you have your. Unless you're a flare. Okay, so we have talked about these 3 domains before, so I'm just going to very briefly go over each. Okay, so we'll start with bacteria. Unicellular prokaryotes that reproduce asexually. Most are heterotrophs. Cyanobacteria are large photosynthetic bacteria that were probably the first organisms to contribute to the oxygen revolution. First organisms to contribute to the oxygen revolution. They are important in ecosystems because they break down organic nutrients. What is beneficial about breaking down organic nutrients? Why is that important? Okay, could be releasing oxygen, except they're heterotrophs, so most are consuming oxygen. Because the nutrients have to flow, right? The nutrients have to cycle back through. So by breaking down organic molecules back down into the monomers, right, then they can be reused again by another living thing. Because those nutrients are going to cycle through. Some bacteria are parasitic and cause disease. And then I would like Slate to explain number 5. You should have no trouble explaining. Go, about their DNA. All right, so let's move on to archaea. And here, these are unicellular prokaryotes that reproduce asexually. They live in what kind of environments? Stream environments. No phototrophs or parasites. Instead are chemoautotrophs. Chemoautotrophs. Now, we haven't done our ecology unit yet, so let me just remind us about what a chemoautotroph. A photoautotroph makes its own, takes inorganic molecules and makes organic molecules using the energy from what? The sun. Chemoautotrophs can take the inorganic and make it organic, right? But they use energy from breaking down other molecules. Okay, so they are chemoautotrophs. They are different from bacteria. Besides the rRNA difference, the cell membrane, they have branched lipids, which allow some to function at high temperatures and different cell walls. They do have some introns, and they have histones like eukarya. What are histones? Histone what? Proteins. Good. DNA wraps around. Good. And then this brings us to the domain eukarya. Unicellular to multicellular organisms, always with membrane-bound nucleus. Sexual reproduction is common, and there are various types of life cycles. There are charts in your book that you can use to compare and contrast the different domains and also the different kingdoms. So let me ask you a couple questions.
Yes.
are maps. Brianna. This is terrifying to me. Okay? Terrifying. Yeah. This is from chapter 1. We are on what chapter right now? 19. What are we getting ready to take this week? Finals. Okay. Some of you said fungi are not eukaryotes. Okay? And some of you said that animals are not eukaryotes. Look at your BioBuddies clicker right now and you give them a smiling poo if they miss that, along with a shame bell. Look at that clicker. There's no shame gongs, gongs of glory. It should be a review. Oh, Trouble in Paradise. Oh, okay. Okay, hopefully you got everything we did so far. It paused just for a minute, so I might have missed part of this section in lecture. If so, I'll re-record it. I don't know why my Facebook account is linked to school and it keeps posting everything. Hopefully nobody puts anything bad. Grace. Okay. Most of you got that. Fungi are not prokaryotic because we just talked about it. Animalia, humans are animals, so are arthropods. And Protista are by and large unicellular eukaryotes. Good job, Kathleen.
That is such a false statement.
I don't know if there's someone.
They ended up getting sex. No one got it right?
No one got it right.
No, one person.
Oh, one person did? You're right. Let's find out who that is. Oh, Gabby! Gabalicious! Well done. One. Yeah, so B, C, D, and E are all characteristics shared by bacteria and archaea. Maybe you want to put this in your notes.
Bacteria?
Yes. So do fungi.
Can I take the photo? Do you take it?
Yes. Remember peptidoglycan? Sure. It's okay.
They have a nucleus, they have fresh ribosomes. So we don't have a nucleus, we do have ribosomes.
I've heard that before.
All right. So we are about ready to transition into our phylogeny talk, so I'm going to give it a little stop right here.